Okay, everybody, here is our PowerPoint on chapters 18 and 20, which is on principles of immunology and antimicrobial drugs. We've talked a little bit about vaccines already, but this is going to be kind of the main focus of chapter 18. So, a vaccine is usually a suspension or a fraction of organisms that's used to induce an immune response. So we've said before that vaccines are like a little bit of the actual organism or a little bit of its antigens or some kind of thing like that. There's a whole different diverse group of vaccines that we're going to list here in just a second. So remember that vacca means cow, which is in honor of Edward Jenner's work with cowpox and smallpox. Use of vaccines is also known as artificial or active immunity, so it's its own little separate part of the immune response. And use of vaccines is really aimed at completely eliminating diseases, especially certain viral diseases. So the different types of vaccines are called attenuated whole vaccines, also known as modified live vaccines. Inactivated whole vaccines, also known as killed vaccines. And then there's also other types of vaccines. Those first two are the two major types. But there's other vaccines that are known as toxoids, subunit vaccines, conjugated vaccines, and nucleic acid vaccines. So really just a huge diverse group here for vaccines. So I thought this table was interesting. This is in your book on page 495, and I did not update this slide. So the one from your new textbook is actually from 2014. But I think that this is actually a really interesting thing to look at, because if you all compare this table from 2004 and then compare it to the one in your book from 2014, I'd be interested to see what differences there are. So I'm going to encourage you all to compare these two. So look at this one from 2004, and then look at the one in your book on page 495, and I want you to compare and look at the differences. And this isn't necessarily something I'm going to really test you a lot on. It's really more so that you are informed, you're educated about the appropriate immunization schedule that's put out by the CDC. And, of course, for your children and your nieces and nephews and your friends' children. And it's just a really great table, really great information about different vaccines and at what age people should be getting those vaccines. So just look this over, compare it to what's in your textbook, the updated one, and I think it's really neat. I could probably look at this for just a long time and kind of think about the differences, think about the changes, and of course also think about some of the controversies. So that brings us to this. So we're not going to really get into this whole big discussion about vaccine use and the pros and cons and the different extreme groups that have kind of cropped up the last few years about the use of vaccines. We're going to really more focus on the technology and the studies that have been done, and just really focus on the science part of vaccines. We could have a debate about vaccine use, and it would go on for hours and hours, I'm sure. So there's still a lot of animal testing being used for vaccines. This is also yet another controversial area, because people who are against animal testing and lab animals being used in, studies for human health. So yet another controversial issue is the animal testing. Another possible future for vaccines is using plants to create new vaccines. The need for more advances in biotechnology to make more effective vaccines without the need for animal testing is huge right now. There's been lots of advancements over the last few years, and those advancements just continue on. New methods of inoculation. So, there's vaccines now that can be breathed in. So instead of having to be given with a needle, or instead of being given orally, there's new vaccines that can just be inhaled. The most important thing for all vaccine use is safety. By far, vaccine safety and studies that prove the use of the vaccine and the safety of the vaccine, absolutely the most important thing is safety and making sure that the vaccine is not only effective, but as little side effects as possible. So, applications of immunology is just talking about a lot of the technology that's used to test for various different things related to health, related to the immune system. By the way, just, feel free to look through this chapter in your book. It has lots of really, really great information about the types of vaccines that are used in the United States. Just so much great information. I mean, information that I couldn't possibly test you on. The test would be, 300 questions long. But really, really good information about different vaccines and a little bit about the history, but mostly about the different diseases. Okay, so another different application, so we're kind of moving on a little bit from the vaccine stuff. The next thing I want to talk about is the monoclonal antibodies, which are antibodies that are developed in lab and can be produced in large quantities, so basically used as medicine. That's what monoclonal antibodies are. And if you want to look at page 502, it basically shows you how the monoclonal antibodies are produced. Immunoelectrophoresis, maybe you guys have heard of electrophoresis before, is when you put an electrical current through various different types of chemicals. Immunoelectrophoresis, the example that you guys want to know for the exam, is called the Western blot test, and that's used in AIDS testing. So you should know that example for immunoelectrophoresis. There's other information in the book. There's information about the details of it on page 279, but you guys don't really have to worry about any of that in terms of the exam. Just know that the Western blot test is used in AIDS testing, and it's an example of immunoelectrophoresis. Agglutination reactions are used quite a bit in various different types of medical tests. So they're very sensitive, very easy to read. Direct agglutination test is something called a titer. Titers are used to basically measure how much immunity you have in your blood against certain diseases, and it can be used to determine whether or not you need a vaccine for it. Indirect agglutination tests are faster. These are some of the strep throat tests, the more rapid strep throat tests. And then the last one is actually used in blood typing reactions. So agglutination of red blood cells is essentially how you can test for blood types, as well as other type like allergy type reactions. Neutralization reaction is used in antitoxin production and study. Complement fixation reactions. If you remember the complement system, I told you guys that I wasn't going to spend a whole heck of a lot of time with it. It has to do with certain proteins that's produced by the liver that's involved in the immune system. If you want more information, it's on page 507, but I'm not going to really test you on this one. Fluorescent antibody techniques are used for testing for certain microorganisms. So obviously, we talked about this before. So exact microorganisms that cause diseases, they have a very specific type of antigen that's on their cells. So this test can actually be used to test for those exact antigens and antibodies, and it basically gives the ability to say exactly what is causing that particular disease. Lots of great information in your book about these tests. They're very helpful in diagnosing diseases. especially if a person is not responding to medication and they're not getting better, then it's a way that we can figure out more about which disease they have. And then you guys maybe have heard of the ELISA test before. It stands for Enzyme-Linked Immunosorbent Assay. It's the most commonly used antibody test. This has just gotten so far advanced, where you can just get to the point where you set up this plate with the samples. You kind of put it into a computer, you push a couple buttons, and it just gives you just a wealth of information. The direct and indirect tests are discussed a little bit more in the book. There is a very detailed diagram on page 510. I certainly am not going to test you on these technologies and the different steps that are involved. The main information that you need is just the information that's found on the PowerPoint. You can almost treat these different technologies as vocabulary words, and just make your flashcards. Make sure you know kind of which ones do what, which ones are for what. The more detailed information about how these tests actually work and kind of the biochemistry involved, that's all going to be in your textbook. For any of you guys that are more interested in it, please feel free to check that out. So I think what I'm going to do now is I'm going to go ahead and end this recording. I have a few minutes left, but it's a good place to stop because this is actually technically the end of the Chapter 18 material. So I'll just pick back up with the Chapter 20 material, and then you'll have two separate recordings for those two separate chapters.